Welcome. This is Lab 6. This is about the genetic control of the pigment called prodigiosin in Serratia strains. Now this follows nicely to Lab 5, the action of genes in biochemical pathways, with a couple of distinct differences here. In lab number 5, we looked at methionine mutants in E. coli. And because methionine was an essential product, we could just see growth or no growth on the plates. Prodigiosin production is not essential to Serratia. So we have to figure out a different way to kind of tease apart the steps in this much more complicated biochemical pathway in a different way that I'll show you in just a moment. So prodigiosin is a bright purple color. And what you need to know that's new here is that when you block a biochemical pathway, oftentimes the product just before the blockage will build up in concentration in the cell. There's nowhere for it to go. And the pathway just feeds into it. It builds up inside the cells, can be exported from the cell, and then diffused through the media. Now, if you have another strain of Serratia close by, it can pick up that intermediate exported from those cells, and it can, if that's useful, in other words, past its biochemical block, it can change the color of that Serratia right at the boundary where it's closer to the exporting strain. Let me show you what I mean on the board here. So what I've got is a very simple biochemical pathway where A, B, and C represent intermediates in the pathway. There's the end product that's non-essential, and the arrows represent enzymes, and of course those enzymes are coded for by genes. So in this case, if we say there's a block between, in other words, there's a mutation in the gene that codes for this enzyme, then this intermediate B is going to build up in concentration. Those are my concentration things there. And that is going to be exported from the cell, and that can be picked up if you streak your plates like I'll show you on this next picture. So this circle represents a petri dish, and C11 is one of our mutant strains, as is OF. Now these strains would be plated out for you. You'd take a toothpick or a swab and pick up a colony and streak it, as I've shown here with the green, and here with the other strain, OF. Now picture the pathway. Each of these is building up an intermediate. It's putting it into the media, and it's diffusing in all directions, but toward this. So in this case, C11 could make a product that allowed OF to complete its pigment pathway. And if it did, then we would see a little color change along this close line. You wouldn't see that color change here. It can't diffuse that far. But right here, there could be a color change that meant that something in C11 was exported, and it allowed OF to complete its pathway, and you detect that completion by the color change you see along that closest line. In lab number 5, the pathway that made methionine was straightforward. It was a series, A to B to C to D to E. Okay? In the prodigiosin pathway in Serratia, it's much more complicated. Not all biochemical pathways are straightforward. Think of the citric acid cycle. Think of other pathways you've seen where there are branches coming together, splitting apart, etc. First of all, I want to convince you that in a straightforward pathway that I'll show you, the phenomenon that you're going to see with Serratia called cross-feeding can't occur. So what I've done is I've put up a pathway here, and there are two of them. And they are to represent two different strains plated on the same plate in the way that you just saw. So let's imagine that this strain is called 1220, and it has a block between intermediates B and C. And that this strain called OF has a block between C and the end product. Now you just saw how in a straightforward pathway like these, the intermediate can build up in concentration and be exported from the cell. So let's now imagine that we're looking at one of those plates where we have 1220 and OF streaked on the same plate. I've shown a picture of that over here. What I want to show you is that there's no possible way, if this pathway was this simple, that these two strains could feed each other. Let's take a look back over here. If we imagine that product B builds up in concentration, is exported, that it could be picked up by the adjacent cells in the bottom streaked strain, and it would provide product B, which could be converted to C, but couldn't be converted to prodigiosin because of the block there. It's easy to see how a one-way, the simple pathway, one could feed the other. If C builds up and is exported, picked up by these cells, the presence of C would allow the creation of that prodigiosin, and so that inner streak would turn the wild type sort of purplish color. But here's the challenge that you face, is that in fact when we do this, we see two feedings. We see that 1220 can make something that can make OF change its color when it's very close to 1220. And OF can make something that can make 1220 on this inner margin change color as well. This phenomenon is called cross-feeding, and the point of this, probably the most challenging lab that you're doing, is to figure out a pathway in which these and all your other results are going to make sense given the blockages in the different strains. Now, here's the deal. Only one block per strain, and that your lab manual will give guidance as to how to think about these more complicatedly shaped pathways, and that'll be you'll have to read and understand that well in order to be able to deduce where all the mutant strains are on this complex pathway.